Turns out, the brain protein that decides if you're immune or inflamed. Your brain is quietly deciding right now whether to tolerate you or attack you. And we just found out that one missing protein, just one, can flip that entire switch. Scientists discovered that when a single molecular gatekeeper disappears, your immune system stops playing by the rules. Your spleen swells, your regulatory immune cells vanish. And deep inside your brain, the protective fatty coating around your neurons starts crumbling. All from one missing piece. So the question is, how many of us are walking around with defects in that exact protein, and nobody knows it yet? For decades, neuroscientists have operated on a pretty straightforward assumption. The brain's immune system, mostly these cells called microglia, should be calm, vigilant but calm. You want them watching for infections and debris. You absolutely do not want them throwing tantrums and torching the delicate infrastructure that lets you think. That balance, that tolerance, seemed to happen naturally. We figured the brain had it figured out. But increasingly, researchers were noticing something weird in genetic studies of Alzheimer's disease and dementia. Certain mutations in a gene called PLCG2 showed up again and again. Some of them seemed protective. Others looked like risk factors. And nobody could explain why one gene would do both. Until this team decided to actually see what happens when that gene completely vanishes. The research comes from a sprawling collaboration led by Gutierrez-Kuri and colleagues, published on bioRxiv in July 2026. This is cutting-edge, unpublished in a major journal work, which means the science is fresh and the implications are still unfolding. But the team that ran this wasn't working in some lab basement. These are serious neuroimmunology people from major institutions, with expertise spanning everything from brain metabolism to immune cell profiling to the actual chemistry of neuronal membranes. They spent months on this, and what they found was unexpected enough that understanding it changes how we should think about brain aging. Here's what they did. They took mice. 3 different versions of them. Normal mice with the PLCG2 gene working fine. Mice missing one copy of the gene, and mice missing both copies entirely. Then they let them grow for 3 months, which is roughly equivalent to a human reaching their early 30s. Then came the invasive part. They cracked open those mouse brains and ran something like genetic archaeology. They looked at the lipids, the fats that make up cell membranes. They measured metabolites, the actual chemical byproducts of cellular work. They sequenced transcripts to see which genes were screaming and which were silent. They even pulled apart the immune cells from the brain tissue and the draining lymph nodes to see who was there and what they were doing. Think of it like taking a photograph of a city, then zooming in on neighborhoods, then on individual houses, then on who lives in each house and what they're arguing about. What they found shattered the assumption that PLCG2 deficiency would just quietly make the brain a little more inflamed. Instead, it did something much weirder. The mice without the gene showed massive immune dysregulation everywhere. Their spleens ballooned, not because more cells were flooding in, but because the cellular composition completely shifted. B cells, which make antibodies and usually help you fight infections, tanked in number. Meanwhile, innate immune cells, the primitive aggressive responder cells, took over. The regulatory T cells, the peacekeepers that normally tell the immune system to chill out, nearly disappeared from the spleen and cervical lymph nodes. Imagine replacing all your diplomats with soldiers. That's what happened. But the brain findings were the real shocker. Brain lipidomics revealed something the team clearly didn't expect. A specific fatty molecule called PIP2, which is like the electrical wiring of cell signaling, became severely depleted. But here's the twist that makes this so strange. Even though the protein was gone, the brain compensated in other ways. The metabolomics showed that cells without PLCG2 actually ramped up oxidative metabolism. They were burning fuel harder and faster, as if the brain was saying, Okay, I've lost my normal signaling pathway, so I'm going to muscle my way through this by working harder. And the myelin, that precious fatty insulation around neurons, started showing signs of damage. Not catastrophic damage, but erosion. The structural integrity of the material that lets electrical signals travel fast was degrading. The proteomics painted an even more detailed picture. Inflammatory markers increased. Proteins related to lipid metabolism shifted. It wasn't a smooth, coordinated response. It was chaotic. It was a brain trying to function while missing a critical piece of infrastructure, compensating frantically, but ultimately unable to maintain homeostasis. And remember, these were young mice, 3 months old, peak of life. If this happens at youth, imagine what accelerated versions of this damage could look like over decades in humans. So what does this actually mean for us? Here's the practical angle. About 1 to 3% of people carry PLCG2 loss-of-function variants. Some of them might notice nothing for years. Others might be at higher risk for autoimmune issues or accelerated cognitive decline. Right now, there's no test, no intervention, no way to know if you're one of them without genetic sequencing. But this study suggests that if you are, your immune tolerance is fragile. Your brain's protective myelin is under slightly more stress than most people's. Over time, that stress could compound. It probably doesn't feel like anything, but biologically you're running closer to the edge. Here's where we need to be careful, though. This is a mouse study. Mice are not humans. Mice live in cages. Humans live in a world of stress, sleep debt, crappy diet, and infections. Mice had 3 months to develop these changes. Humans would be developing them over 3 decades. We don't know if the changes scale linearly. We don't know if there are checkpoint genes that kick in and protect human brains that don't exist in mice. And this was published on bioRxiv, which means it hasn't yet survived the peer review crucible. It's probably solid. The data looks comprehensive, but it's not locked in stone yet. What's genuinely fascinating is what this research reveals about the broader logic of immunity and aging. For years, neuroscientists thought brain inflammation was the root of cognitive decline. And maybe it is, but this study suggests the real problem might be the opposite: loss of tolerance, loss of the cellular mechanisms that keep immune aggression in check. Your brain gets inflamed not because inflammatory cells are aggressive, but because the inhibitory brakes have failed. That reframes Alzheimer's, neurodegeneration, and aging itself as tolerance diseases, not inflammation diseases. The best therapy might not be killing inflammatory cells. It might be rebuilding the regulatory architecture that tells them when to stop. This is the kind of finding that sits quietly in the literature for 2 years, then suddenly spawns 15 follow-up papers and a whole research direction. Someone's going to ask whether PLCG2 variants predict cognitive decline in humans. Someone's going to try to rebuild PIP2 in the brains of these knockout mice and see if it helps. Someone's going to look at dementia patients and check their PLCG2 status. And that's how science actually moves. Not with one spectacular discovery, but with one weird finding that makes someone curious enough to dig deeper. Your immune system and your brain are in constant negotiation about whether you're a friend or a threat to yourself. One missing protein erases the immune system's ability to listen to reason. Turns out, scientists have been busy, and they're not done yet.